Okay, so as I said, I think the FRQs this year were really straightforward. There were a couple errors that I saw on their like naming conventions for like using the bold words and stuff. But maybe they're switching stuff up. We'll just have to adapt to that. But we're going to start with question 1 here. Question 1 was this one about auxins. It says auxins are plant hormones that coordinate several aspects of root growth and development. Indoleacetic acid, IAA, is an auxin that's usually synthesized from the amino acid tryptophan in figure 1 here. The gene TRPT encodes the enzyme that converts tryptophan into indole 3 pyruvic acid I3PA, which is then converted into IAA by the enzyme encoded by the gene YUK. Okay, so that's this thing here. That's this, oops, can't see that for some reason. This question. And it says, for question 1, it says, circle one arrow that represents transcription of on the template pathway. Identify the molecule that would be absent if the enzyme YUK is non-functional. Okay? So, both this arrow and this arrow both represent transcription, right? So you could have put either one. Probably this one is fine. That's what most people are going to put because you were already talking about YUK here. Okay, so this one is the one that you should have circled, so that would be 1 point. Okay, then it says the second part of the question was identify the molecule that would be absent if the enzyme YUK was non-functional. Well, if this enzyme YUK is non-functional, that means that you don't get to go through this arrow, and if you can't go through this arrow, you will not make IAA, right? So you wouldn't make IAA, and by saying you can't make IAA, that's the second point. Okay. Part B, predict how a deletion of one base pair in the fourth codon of the coding region of the gene TRPT would most likely affect the production of IAA. Justify your prediction. Okay, so we got to predict and to justify. That's 2 points. We know that anytime that there's a deletion, you're going to end up with a nonsense mutation because of a frame shift, right? And so that is going to then significantly decrease or stop the production of IAA, because if you can't produce TRPT, right, then you can't make I3PA, and if you can't make I3PA, you can't make IAA, right? So that's 2 points. 1 point for saying that it's a nonsense mutation, 1 point for saying that you can no longer make IAA. Explain one feedback mechanism by which a cell could prevent production of too much IAA without limiting the production of I3PA production. Okay, so basically what that means is we need to limit this without limiting this. Okay, and what we need to do then is we need to affect this part of the production pathway then. And so generally speaking, what we're going to do is we're going to have IAA come back and inhibit enzyme YUK, right? And so if IAA inhibits enzyme YUK through some sort of feedback mechanism, negative feedback, allosteric inhibition here, that would be probably the best answer for that, right? So that was how many points? Explain, so that's 2 points for explaining there. So boom, boom. The next question was, rhizobacteria are a group of bacteria that live in nodules of plants. Rhizobacteria can produce IAA and convert atmospheric nitrogen into forms that can be used by plants. Plants release carbon-containing molecules into the nodules. Based on this information, identify the most likely ecological relationship between plants and rhizobacteria. Well, you don't really even need that information because this is something that we studied verbatim in here. This is like the mutualism example that we always use. Nitrogen-fixing bacteria in the root nodules of plants. Right, so it's mutualism, 1 point for saying mutualism. Describe one advantage to the bacteria of producing IAA. Well, it says that IAA is responsible for coordinating several aspects of root growth and development. Okay, so if you produce IAA and that makes the roots grow more, that's more places to live for the bacteria. So they're going to be able to increase in population size based on producing that IAA. Again, so 1 point there, 1 point there. We are up to a total of 8 points. So we need 2 points from the remaining part. The remaining part says, a researcher removed plant nodules and identified several cheater rise of bacteria that do not produce IAA or fixed nitrogen. Describe the evolutionary advantage of being able to of being a bacterial cheater in a population composed of predominantly non-cheater bacteria. Okay, so all the non-cheater bacteria are getting the plant to give all of this carbon because they're producing all the nitrogen, right? So they're in that mutualistic relationship. But the cheater bacteria, they don't spend any energy doing nitrogen fixation. So they still get all the benefits, which is the carbon, but they don't have to do any work, which is producing all of the usable nitrogen, right? So they get to use that energy for reproduction, and therefore they would then outcompete the non-cheater bacteria. Predict the change in the bacterial population that would cause the plant to reduce the amount of carbon-containing molecules provided to the nodule. Okay. Well, if there was a change in population where the cheater bacteria became more prevalent than the non-cheater bacteria, then that would reduce the amount of nitrogen being produced, and then that would reduce the amount of carbon-containing molecules that are provided to that root. Right. So again, that is 10 points there. All 10 points are accounted for. Questions on question 1? Okay, question 2. 10 points. Question 2 says, a student studying two different aquatic plant-eating unicellular protists, species A and species B, designed an experiment to investigate the ecological relationship between the two species. That's what we see in table 1. In the treatment group 1, student placed 10 individuals of species A into a container with a liquid growth medium and 10 individuals of species B into a separate container with an equal amount of the same liquid growth medium. In treatment group 2, the student placed 5 individuals of each species into a single container with a liquid growth medium. The student then maintained the containers under the same environmental conditions and recorded the number of individuals in each population at various time points. The results are shown in table 2. Cool. The growth curves for species B in group 1 and for species A in group 2, the shaded columns, have been plotted on the template. Use the template to complete the appropriately labeled line graph to illustrate the growth of species A in treatment group 1 and species B in treatment group 2, the unshaded columns. Okay, so what we have here, oops, that's not it. What we have here is a graph. Right? And you just needed to plot this. So the very first thing that you should have done is you should have determined what these axes are, right? So down here on the bottom, this is the independent variable, which is going to be time in hours. So you should have written time, a little lower. You should have written time in hours, like that. And then you should have determined what the increments were. And so you see that species B group 1 levels out about right here, right? And so it levels out at 700 at hour 50, right? So we know this is hour 50, which means that this must be hour 25, right? Which means that these are in 5s, right? And then it levels out at 700. So we know this is 700 here, so this must be 500, and this must be 1,000. And then that on the side was the number of individuals. Does it say anywhere what that is on the side? Number of individuals in each population. Okay. So this on the side of that then should have been number of individuals. And then at the top, you should have written something like individuals in each protist population in 2 treatment groups. Okay, something like that. And then you should have gone through and plotted them. I'm not going to go through and plot them. That's a waste of time, I think. but you should have just plotted them on here. So you'd go and you'd say, okay, 0, 10, 10, 100, 20, 400, yada, yada. And then you do connect the dots that in that case, because these ones have the dots connected, right? So that would be, let's see, complete. Complete's a weird word to have bolded. So let's hold off on figuring out how many points this is, because we don't know right now. Okay. As shown in the table, the student establishes treatment group 2 with 5 individuals of each species. Provide reasoning for the reduced initial population size. Okay. Well, in treatment group 1, they used 10 individuals per container. And so in treatment group 2, they used 10 individuals, but they have 2 different species, so it has to be 5 per species. This maintains the same initial population density, right, or initial species density. Rather, it's 2 different populations, but we are guessing that they do share carrying capacity, and so they want to have that initial species density be the same. The student claims that species A and B compete for the same food source. Provide 2 pieces of evidence from the data that support the student's claim. So on the graph here, you would be able to see that in group 2, when they're grown together, that as species A increases, when they get to a certain point, species B has to start decreasing, right? So when they get to this point where this is 50 and 100, right? This one goes up from 100 to 250, but this one goes down from 50 to 25, indicating that they are competing with one another for resources, right? And you can see when they are grown separately, that doesn't happen. When they're grown separately, their populations continue to increase, right? Up to a certain point. Predict 2 factors that will most likely limit the population growth of species A in treatment group 1, right? So we can see here that they get to the end, and I think you could have picked any density dependent factor here. So you could have said that it was, lack of food. You could have said that it was lack of physical space. You could have said that it was buildup of toxic waste. Probably less so disease, right? But those 3 things would have been good. I also think less so food, right? Because if it didn't mention in the experiment that they gave them any more food. It said that they just grew them in an initial amount of growth medium. And so once they got to this point where they leveled out at 1500, you would have seen their population decline as they then couldn't support that population anymore because they didn't have any food. I don't know if College Board's going to be a stickler on that. I would guess no. They'll probably accept any density dependent factor here, so food, toxic waste, or space. Many protists contain an organelle called the contractile vacuole that pumps water out of the cell. The student repeated the experiment using a growth medium with a lower solute concentration. Predict how the activity of the contractile vacuole will change under the new experimental conditions. Well, if the new growth medium has a lower solute concentration, that means it is more hypotonic to the cell. And so if the cell is hypertonic, more hypertonic now, that means that water is going to diffuse into the cell, and if water diffuses into the cell, it's going to explode unless the contractile vacuole is pumping it out. So the contractile vacuole needs to increase its activity in order to pump the water back out of the cell, right, so that it doesn't explode. So we would say that the activity of the contractile vacuole will increase because more water will diffuse into the cell via osmosis, and so water is going to need to be taken out. Okay, so let's look at the points here. We got to justify and a predict. Those are 2 points. We're pretty sure about that. So 1, 2. Then we got predict 2. But then above this, we see provide 2. But the 2 is bold here, but it's not bold here, but it's capital in both cases. What's the difference? I have no idea. I think this was an error. But let's just guess that predict capital 2 means that they're going to give you a point per. Maybe. Okay. So we're gonna write that. Just say a point per. Boom boom. Okay. Then we are going to say provide 2 pieces of evidence. Right. So again, we're going to give 2 here. I don't know what the difference is between the bold 2 and the not bold 2, but we're going to go 2. Okay. Then we've got provide reasoning, so that's just 1. Right. So that gives us a 7. So that must mean that the complete, the graph part, that must have been worth 3. right? I would guess that that means that it's going to be 1 point for each of the lines that you drew, and then 1 point for correctly labeling the graph. Just a wild guess, but again, I think College Board was a little bit crazy with some of their capital words and bold words that didn't really line up the way that you would expect. This mistake. Yep. At the end of the period, I can. Great. Right. Okay. Question 3 was the pyruvate dehydrogenase complex, or PDC, catalyzes the conversion of pyruvate acetyl-CoA or pyruvate into acetyl-CoA, a substrate for the Krebs cycle. The rate of pyruvate conversion is greatly reduced in individuals with PDC deficiency, a rare disorder. Identify the cellular location where PDC is most active. Okay, well, in order to do that, we need to know about cellular respiration, right? And so we know you get pyruvate at the end of glycolysis, which happens in the cytoplasm, right? And then you pump this pyruvate into the mitochondrial matrix. So the answer that they're looking for here is mitochondria, or perhaps mitochondrial matrix, if they're going to be a stickler for that. But I also think that they need to give points for cytoplasm, right? Because they didn't say anything about this being a eukaryote or a prokaryote. And so in a prokaryote, PDC would be active in the cytoplasm, not in the mitochondria, because they don't have mitochondria, right? So I think they need to give points for both cytoplasm and mitochondria. Maybe they will be a stickler and say that it has to be mitochondrial matrix, but hopefully not. The next part of the question says, make a claim about how PDC efficiency affects the amount of NADH produced by glycolysis and the amount of NADH produced by the Krebs cycle in the cell. Provide reasoning to support your claims based on the position of the PDC catalyzed reaction and the sequence in the cellular respiration pathway. Okay, so if we said that PDC happens right here, or occurs somewhere in this part here, all right, then glycolysis, which is before that, would be unaffected, right? Actually, well, here's your acetyl-CoA. I don't know why that's getting all crazy, but here's acetyl coenzyme A, right? So it's there. So glycolysis is unaffected. Krebs cycle, though, is affected. So Krebs cycle would have reduced NADH production, and then glycolysis would have the same NADH production, right? Let's tally the points, and we'll see why this is kind of weird. Maybe not so great. So if we go back and we say make a claim and, see this capital and? That tells us that we should get a point for making a claim about the first one and making a claim about the second one, right? And provide reasoning. So that's 3 points, right? And then the next point, next part C, calculate, that should be another point. But that's 4 points, which means that A wouldn't be worth any points because this question is only worth 4 points. So I think they misused this and, this capital and here, and this and should have been a lowercase and. Based on scoring conventions from previous years, that and should have been lowercase, meaning that you only get a point if you make a claim about both the Krebs cycle and the glycolysis. You don't get separate points for those. So again, identify is 1 point. That was the identify where it's going to happen. 1 point for saying what's going to happen in glycolysis versus Krebs, and then 1 point for providing reasoning for that. Wait. 1 point identify, 1 point for identifying glycolysis and Krebs, and then 1 point for providing reasoning, right? And then the last one was a calculate, which again, this is not the best use of terms here. Calculate, we know, should be 2 points, and it should be 1 point for showing your work and 1 point for giving the correct answer. But there's only 1 point available here, and the reason why they're only going to give 1 point for this is because you don't need to show work for this. It says PDC deficiency is caused by mutations in the PDHA1 gene, which is located on the X chromosome. A male with PDC deficiency and a homozygous female with no family history of PDC deficiency have a male offspring. Okay? So that means that the female is X normal, and the male is X deficient Y. Okay? Calculate the probability that the male offspring will have PDC deficiency. Well, the male offspring is getting the Y from the father and the X from the mother, right? And so that means that he has a healthy X, and the Y doesn't carry the gene at all, right? So 0%. The reason why you're not going to get a point for the calculation in that case is that there is no calculation. It's just a Punnett square maybe, but you probably didn't even need to do a Punnett square based on, you know, the fact that it was a pretty simple calculation. So 1 point for that. It's just going to say 1 point for giving that answer, which is 0%, right? And the reason why I don't like that is because that it just giving a point for the answer, giving a point for saying something, that is not calculate, that is identify. So it should have, based on their scoring here, it should have said identify the probability, right? That would have made more sense. All right. Let's go to number 4. Number 4, acetylcholine is a neurotransmitter that can activate an action potential in a postsynaptic neuron, figures 1 and 2. A researcher is investigating the effect of a particular neurotoxin that causes the amount of acetylcholine released from the presynaptic neurons to increase. Okay, so basically what's going to happen is this presynaptic neuron is going to increase more acetylcholine into the synapse. Okay, that's going to bind to these receptors, and it's going to cause the action potential to happen. So, for question 1, which says, describe the immediate effect of the neurotoxin on the number of action potentials, increases, right? The number of action potentials will increase because the amount of acetylcholine is going to increase. That's 1 point. Predict whether the maximum membrane potential of the postsynaptic neuron will increase, decrease, or stay the same. This is a chemical synapse here, so there's nothing to indicate that you're going to see any hyperpolarization or anything like that. So it's just going to stay the same, right? I think that's the best answer in this case. It doesn't indicate anything other than that. If it were a electrical synapse, that might be something different, but it's not. It hasn't indicated anything like that. So we're just going to say it's going to stay the same. So 1 point for saying that the number of action potentials increases. 1 point for saying that the maximum membrane potential, this spot right here, would stay the same. Okay. The researcher proposes 2 models, A and B, for using ACHE, an enzyme that degrades acetylcholine to prevent the effect of the neurotoxin. In model A, ACHE is added to the synapse. In model B, it's added to the cytoplasm. Predict the effectiveness of each. Again, this is an incorrect use of capitals here because this question is only worth 4 points, and there's 4 bold words. So you can't have an each here because that would tell you that predict should give you 1 for predicting each thing, but it's just going to be 1 point for predicting both things, right? And then providing reasoning. So 1, 2, 3, 4 there. Okay, so if we add this enzyme that degrades it into the synapse, right, that's going to take away these things, which would then be effective at stopping the neurotoxin, at preventing the effects of the neurotoxin, because as the acetylcholine gets into the synapse, it gets degraded by the ACHE, right? If you add it to the cytoplasm of the postsynaptic neuron, nothing happens because this acetylcholine never gets into here. It stays in the synapse, right? So there's your provide reasoning and that stuff. 4 points there. Question 5. A researcher studying the evolutionary relationship among 5 primate species obtained data from sequences of mitochondrial DNA from a representative individual of each species. The researcher then calculated the percent divergence in the sequences between each pair of primate species. Okay? Based on fossil data, the researcher estimates that humans and their most closely related species in the data set diverged approximately 7 million years ago. Using these data, calculate the rate of mitochondrial DNA percent divergence per million years between humans and their most closely related species in the dataset. Round your answer to 2 decimal places. Again, this is another, I think, misuse of the term calculate, because calculate should be worth 2 points, and it's only worth 1 point in this case because this is a 4-point question with 4 bold words. So each one must be worth 1 point. Okay? Now, how do you calculate this? Well, you find the most closely related species. So we got humans and chimps, 8.8% divergence there, and they had 8.8% divergence in 7 million years, right? So that'll give you the percent divergence per 1 million years if you do 8.8 divided by 7. Now, I think the reason why they're only going to give 1 point for this calculate, even though there is a calculation combined, or calculation involved in this, is that this final answer here, this calculation, could also be a final answer, because on AP tests, it has been traditionally accepted to give improper fractions as your final answer. So you probably could have just given the final answer 8.8 divided by 7, which is also the calculation. So there was no other calculation. So it wouldn't have made sense to give a point for a final answer and the calculation if they were the same thing. But again, I think it should have just said identify. But that's all right. So that's 1 point for that. Point tally. 1 point. The next part of the question says, using the data in the table, construct a cladogram on the template provided. Provide reasoning for the placement of gibbons as an outgroup in the cladogram. So I'm going to make another statement about this. I don't love that this is a cladogram. All the cladograms that we've studied in the class, of course, have derived characters on them. This one is a one that's using mitochondrial DNA differences, which you would then, based on part A, you would have been able to estimate the time period by which they diverged. So it would have made much more sense to use a phylogenetic tree here rather than a cladogram, because you can have that relative time length on it. And also there's no derived characters on here. But again, we're just going to let it slide. We're going to do what they want us to do here. And so basically you're just going to go through and you're going to figure out which things have the least amount of differences. So if you put humans at the top, you'd put chimps here because they have that 8.8, right? Then the next one would be humans and gorillas, right? And so the gorillas would go here. Whoa. And then you'd have orangutans. And then gibbons. Right? And then it said provide reasoning for the placement of gibbons. Well, the gibbons have the greatest amount of divergence from all the other species, and so they should be the outgroup in this cladogram. Okay? So that was construct and provide reasoning, so that should be 2 points. 2 more points there. Boom. Boom, boom. And then the last part says, in the cladogram, draw a circle around all the species that were descended from the species indicated in the node within the square. Again, this makes much more sense to do this on a phylogenetic tree rather than a cladogram. But again, we're just going to roll with it. So we would say that the gorilla, the chimp, and the human all were descended from this node here. 1 point for that. Questions on question 5? Okay. Question 6. Question 6 says, there's this yeast, and it's a single-cell organism. Amino acid synthesis in yeast cells occurs through metabolic pathways, and enzymes in the synthesis pathways are encoded by different genes. The synthesis of a particular amino acid can be prevented by a mutation of a gene encoding an enzyme in the required pathway. Okay. A researcher conducted an experiment to determine the ability of yeast to grow in medium that differ in amino acid content. Yeast can grow as both haploid and diploid cells. The researcher tested 2 different haploid yeast strains, mutant 1 and mutant 2, each with a single recessive mutation. I think this part's going to be important, that it says with a single recessive mutation, for reasons we'll talk about in a second. And a haploid wild-type strain, the resulting data are shown in Table 1. Identify the role of treatment 1 in the experiment. So in treatment 1, we can see that we gave them all the amino acids. Okay? So we give them all the amino acids and they all grow in it. Basically what you're trying to determine here is, can these things grow? Right? If we're just growing them normally, are they able to grow? And yes, they are able to grow. And so we call that the control. Right? This is the control to make sure that even if we don't change anything, even if we don't mess with the amino acids, they can all grow. All right. So that's 1 point. The next part says, provide reasoning to explain how mutant 1 can grow in treatment group 1, or treatment 1 medium, but can't grow in treatment 3 medium. Well, if we look, treatment 1 has all the amino acids, but treatment 3 doesn't have methionine. And so that would indicate to us that mutant 1 does not have the ability to synthesize methionine. Right? Boom. Last point in this one says, yeast mate by fusing 2 haploid cells to make a diploid cell. In a second experiment, the researcher mates mutant 1 and mutant 2 haploid strains to produce diploid cells. Using the table provided, predict whether the diploid cell will grow on each of the 4 media. Use a plus sign to indicate growth and a minus sign to indicate non-growth. I think that there are 2 answers to this. Okay? If you answer the question based on the prompt, which said that mutant 1 and mutant 2 have a single mutation only, right? That means that mutant 1 combined with mutant 2 then, as both haploid, single recessive mutations, they would both be heterozygous for this gene, and therefore they would be exactly like the wild type. So we should put plus, 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 plus. I think that's answer 1. I also think that based on what they want you to be able to do in this class, which is analyze scientific data, I think they need to also accept plus, minus, plus, plus. Because if we know that mutant 1 and mutant 2 couldn't grow in this treatment group 2, sort of by their powers combined, they still can't. Right? So again, this plus, 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 plus is the most correct of all of the answers. But I also think that College Boards should accept plus, minus, plus, plus, because in this case, based on the data provided in the table, mutant 2 and mutant 1 and mutant 2 cannot grow in treatment group 2, and so even if you combine them, they still shouldn't be able to grow. Again, that's a little bit weird. And the last point, question 7. Question 7 you can see here is also 3 points. And in question 7, you can see that it says, a researcher is studying patterns of gene expression in mice. The researcher collected samples from 6 different tissues in a healthy mouse and measured the amount of mRNA from 6 genes. The data is shown in figure 1. Okay? Based on the data provided, identify the gene that is most likely to encode a protein that is an essential component for glycolysis. Provide reasoning to support your identification. Okay? How important is glycolysis? Pretty important. So which cells can survive without glycolysis? None. So we need it in all the cells. So it's got to be gene G because it's in all the cells. Okay, so identify there. That's 1 point. Then point 2 says provide reasoning to support your identification. We just kind of did that. We said that since glycolysis is something that has to happen in all cells, that we would expect gene G to be present in all cells. Point 2. Okay, the last point is the researcher observed that tissues with high level of gene H mRNA did not always have high gene H protein. Provide reasoning to explain how tissues with high gene H mRNA have no gene H protein. So any post-transcriptional regulation here would have been okay. So if you said microRNA interference, if you said repressor proteins on the mRNA, if you said that the proteins were being produced but then they were being destroyed by enzymes in the cell, if you said that there was a mutation, it could have been a mutation in gene H that caused the start codon to be not non-functional, right? You could have said that there would have to be another thing around in order to create gene H protein, a secondary mRNA. You can put those up here. Any of those things would have been fine as long as it was, scientific and logical. Those would all be fine. Okay? And then the last question, 8, was... The petal color of the Mexican morning glory changes from red to blue, and the petals swell, or petal cells swell during flower opening. The pigment heavenly blue anthocyanin is found in the vacuole of the petal cells. Petal color is determined by the pH of the vacuole. A model of the morning glory petal cell before and after flower opening is shown in Table 1. Identify the cellular component in the model that is responsible for the increase in pH of the vacuole during flower opening, and describe the component's role in the changing pH of the vacuole, or changing the pH of the vacuole. So if we look here, first thing that we need to know is that the pH is the measure of H plus ions. And if the pH goes up, that means that the H plus ion content is going down, right? And so it says identify the cellular component that is responsible for increasing the pH. That means that the cellular component that is causing the pH to go up is the one that is allowing H plus ions to leave, which is this thing, the K plus H plus transport protein, right? And describe the component's role in changing the pH of the vacuole. Okay, well, that's where the H plus ions exit, right? And why does the H plus ion exit through this point? Well, it's using it as a co-transport. As the H plus ion goes out, the K plus ions can go in, and as the K plus ions go in, that's going to increase the K plus ion concentration in here. So the H plus ions go down their gradient, and the K plus ions go up their gradient. So 2 points there. And the last point was 4. It says, the researcher claims that the activation of the K plus H plus transport protein causes the vacuole to swell with water. Provide reasoning to support the researcher's claim. Well, if we are increasing the amount of K plus ions in the vacuole, right, that means that we are increasing the solute potential in there. And if the solute potential increases, that means that the osmosis, the osmotic pressure, is going to increase and water is going to diffuse into the vacuole, which it will then cause it to swell with water, right? So increasing the amount of K plus ions increases the amount of osmosis, causing it to swell, right? And that's your last point on the FRQs. Anybody have any questions on any FRQs? Anybody have any